Polygenic score is the combined measure of a larger number of risk genes for any disease. In normal population studies, polygenic score for alcohol dependence has been found to be negatively associated with cognitive function. No evidence was found for a causal association of cognitive impairment for RS1229984 in alcohol dehydrogenase 1b or RS671 in alcohol dehydrogenase 2 in the normal population. Light to moderate alcohol use has been found to be associated with a decreased risk of cognitive impairment in the normal population. However, this J-shaped relationship between alcohol use and cognition could be attributed to potential abstainer errors and reverse causality bias. Alcohol use polygenic score has been found to be associated with alcohol use, alcohol-related morbidity, and all-cause mortality. Polygenic score for alcohol use disorder has been found to be positively associated with alcohol use. Polygenic score for the problem subscale of alcohol use disorders identification test has been found to be associated with alcohol use disorder. Polygenic score for drinks per week has also been found to be positively associated with drinks per week. The study design was a cross-sectional study of a representative sample of people with psychotic disorders encompassing the whole of Finland. The super study aimed to collect genotype and phenotype information from at least 10,000 Finnish patients suffering from psychotic mental illness. A DNA sample acquired via phlebotomy from every study subject went through whole genome sequencing. The genomic data were then be analyzed in association with the phenotype data collected. The pilot study was commenced in the fall of 2015 and ended in 2016. The main study started in 2016 and continued for 3 years. 1. Being less than 18 years. 2. Being greater than or equal to 70 years. 3. Being unable to provide informed consent as evaluated by the trained research personnel or attending physician. 4. Not living independently. 5. Missing information on education. 6. Missing information on depressive symptoms. 7. Missing information on hazardous drinking polygenic score. About 54% of participants were males and 46% females. The mean age was 45 years for males and 47 for females. The mean age of onset was 26 years for males and 27 for females. About 1/3 of the males and 2/5 of the females had the highest basic educational of 12 years. About 90% of males and 75% of females were living without a spouse. About 2/3 of participants had depressive symptoms. Most of the participants were on psychotropic medication. About 95% of them were taking antipsychotics, 25% benzodiazepines, about 1/3 antidepressants, and about 1/5 mood stabilizers. Hazardous drinking polygenic score was 7.95 times 10 to the negative 7th power for males and 7.63 times 10 to the negative 7th power for females. About 1/3 of participants were males and 2/3 females. The mean age was 42 years for males and 43 for females. The mean age of onset was 30 years. More than 2/5 of participants had the highest basic educational of 12 years. About 4/5 of males and 2/3 of females were living without a spouse. More than 2/3 of participants had depressive symptoms. Most of the participants were on psychotropic medication. A total of 93% to 94% of them were taking antipsychotics. About 1/3 benzodiazepines, 1/3 males and 2/5 females were taking antidepressants, 2/5 males and 1/3 females were taking mood stabilizers. Hazardous drinking polygenic score was 7.71 times 10 to the negative 7th power for males and 7.77 times 10 to the negative 7th power for females. Hazardous drinking polygenic score was not associated with visual memory and reaction time decline in schizophrenia and schizoaffective in outpatients below 70. Adjusted with age, age of onset, education, household pattern, and depressive symptoms when a reaction time test and a memory test were used without an assessment of executive functioning and without correcting for manic symptoms. Whereas hazardous drinking was associated with better cognition in our previous studies with the same population. It is still unclear whether hazardous drinking polygenic score is associated with global cognition decline.